Now that we have a greater understanding of the properties of cardiac tissue, we can build upon that as we learn about the electrical events of the heart. So the heart can depolarize, that is, it can change its cell resting membrane voltage from rest, which is typically about minus 90 millivolts, and the voltage can increase towards zero. This depolarization will cause the heart to contract, and this depolarization does not require stimulation from the nervous system. That is, the heart muscle tissue itself is able to depolarize in a rhythmic fashion. However, we do see that the heart rate as well as the force of contraction does have the ability to be altered by the nervous system, specifically the autonomic nervous system. The sympathetic branch of the autonomic nervous system will tend to increase heart rate as well as increasing the force of contraction. On the other hand, the parasympathetic division of the autonomic nervous system will tend to slow down heart rate. Approximately 1% of cardiac tissue is a specialized tissue that is called myocardial conducting tissue. This is tissue that is similar to the nervous system tissue and is described as what we call autorhythmic. So that is, it is able to set a baseline frequency for its depolarization, and that causes a rhythmic contraction of the heart as a result of that. So it's these myocardial conducting tissue structures that make up what we call the intrinsic conduction system of the heart. We have just learned that the cardiac muscle tissue has the ability to depolarize itself and set a baseline rate of depolarization as well as a baseline rate of contraction for the heart. However, we also described the fact that the nervous system does have control over the pacemaker cells that sets the pace of the heart. Specifically, the parasympathetic division of the autonomic nervous system can function to decrease heart rate, and these signals are sent to the pacemaker cells of the heart through the vagus nerve, which is one of the cranial nerves. The sympathetic division of the autonomic nervous system can increase heart rate as well as increasing the force of contraction of the heart. These signals are also delivered directly to the pacemaker cells of the heart through the sympathetic nerves that emerge from the vertebral column and from the spinal cord. So myocardial conducting cells have an unstable resting membrane potential, which constantly drifts toward action potential threshold. So recall that neurons in the nervous system require a stimulus in order to bring the membrane voltage up to that threshold voltage, and it's at that threshold voltage that those neurons will fire an action potential. Cardiac pacemaker cells, because they have an unstable resting membrane potential, do not need a stimulus. That is, after a short period of time, the membrane voltage will reach the critical value of threshold voltage, and an action potential will fire as a result of that. So this means that the cells have the ability to depolarize on their own, as well as fire action potentials without nervous system input. So of course, as we had just seen in the previous few slides, the nervous system can and does influence heart rate in specific situations. The intrinsic conduction system by itself will primarily set the baseline heart rhythm, and it's the nervous system that has the ability to modify that baseline heart rhythm depending on the situation. The pacemaker potential is the first part of this graph, so it is the part that is below the dashed line. We see that the pacemaker potential slowly climbs up towards the threshold voltage, and we see this at part 1 in the graph towards the left. So as the pacemaker potential increases towards that threshold voltage, what's happening is that sodium channels are opening, and this is allowing sodium, which is a positively charged ion, to trickle into the cardiac pacemaker cells, causing the membrane voltage to slowly rise towards that threshold level. So once the membrane potential reaches threshold, we see that the myocardial conducting cells will then depolarize very rapidly, which is shown as the steep uphill at item number 2 in this graph. And this depolarization is caused by calcium, which is also a positively charged ion that's entering the cell through calcium channels. After a short time, the calcium channels will become inactivated, and calcium will no longer be able to enter the cardiac pacemaker cells. Simultaneously, potassium channels will open and allow potassium, which is once again also a positively charged ion, to exit the cells. And this is going to bring the membrane voltage back below threshold during the repolarization phase of the graph, which is shown in part 3, or item 3. So the intrinsic conduction system is composed of the following structures. The first is the sinoatrial node, which is also known as the SA node, located in the upper part of the right atrium. It generates impulses or depolarizations at a frequency of about 75 times per minute, which is approximately one impulse every 0.8 of a second, or every 0.8 seconds. The second structure is called the atrioventricular node, which is located in the lower aspect of the right atrium, in close proximity to the right atrioventricular valve. Connected to the atrioventricular node is a bundle that is very appropriately called the atrioventricular bundle. It's also called the bundle of His, and this structure is found in the upper portion of the interventricular septum. The AV bundle, or the atrioventricular bundle, as it continues down towards the apex of the heart, will branch into two branches, which we call the left and the right bundle branches. As the bundle branches then fan out through the right and left ventricle, that is through the myocardium of the right and left ventricle, they are then identified as the right and left subendocardial conducting network, which are also called the right and left Purkinje fibers. The sinoatrial node sets a rhythm that's known as sinus rhythm, and this is the rhythm that sets the pace for the entire heart itself. And so for this reason, the sinoatrial node is often referred to as the pacemaker for the heart. Sinus rhythm is, on average, a resting heart rate of 75 beats per minute. So that is, the sinoatrial node fires at an average of about 75 impulses or action potentials per minute in order to get the heart to contract. So the SA node is what stimulates the atria to contract, and the SA node is also what stimulates the atrioventricular node to fire an action potential. And the atrioventricular node is the node that is going to stimulate the ventricles to contract. We see that there is a brief delay between when the AV node is stimulated and when the AV node actually depolarizes. And this delay is typically about 1/10 of a second, or 0.1 seconds. And what this does is it gives time for the atria to contract and push their blood into the ventricles before the ventricles begin to contract. In total, including the 0.1 second delay between the atrioventricular node and the stimulation of the atrioventricular node and the atrioventricular node depolarization, a sinoatrial node impulse to the depolarization of the ventricles takes under just about a quarter of a second, which clocks in at about 0.22 seconds on average. As mentioned previously, what happens is that the delay between the depolarization of the atria and the depolarization of the ventricles is going to give time for the atria to fully contract and allow the ventricles to fill properly with blood that is being pushed from the atria. What are possible defects that are seen in the intrinsic conduction system? These defects include things like arrhythmias. Arrhythmias are irregular electrical activity within the heart. And we also see fibrillations, which are rapid and out-of-sync contractions, which is usually due to the disruption of the SA node by typically rapid activity in other regions. And usually this rapid activity results to rapid contractions in other regions of the heart. In particular, ventricular fibrillation causes the ventricles to become useless as pumps because it results in a stoppage of blood circulation because, as we saw in previous modules, the ventricles are the chambers that are going to be pumping blood into the body's major arteries. And so if blood is not moving out of the ventricles, it's also not moving into the arteries as well. Defibrillators are devices that depolarize the entire heart, and they do this in the hope that normal sinoatrial node function will resume. So essentially it's kind of a reset button for the SA node, and as we've just seen, the SA node is what stimulates the atrioventricular node as well. So by resetting that SA node, we can reset the rhythm for the entire heart and prevent it from fibrillating. Electrocardiography or electrocardiograms measure electrical currents throughout the heart, so that is all of the depolarizations and repolarizations that are occurring as the heart electrical activity changes. Electrocardiograms, which are usually abbreviated ECG or EKGs, are the graphic recordings of these electrical currents and these electrical changes during the cardiac cycle. So here is a graph of a typical electrocardiogram from a healthy individual. Starting from the left side of this tracing, we'll focus on three structures, in particular what are called three waves or deflections: the P wave, the QRS complex, and the T wave. The P wave is the deflection that corresponds to the depolarization of the atria. So both the left and the right atria will contract sometime after the P wave but before the QRS complex. Note here that the electrocardiogram is an electrical recording. So all we can see is the electrical activity. We can't actually see the contractions of any of the musculature of the heart. Next, we have the QRS complex, which represents the depolarization of the ventricles. So both the right and the left ventricles are going to contract sometime after the QRS complex but before the T wave. And finally, the T wave represents the repolarization of the ventricles. The depolarization of the atria, which is going to occur by stimulation from the sinoatrial node, will create the P wave in an electrocardiogram. Atrial contraction will immediately follow the end of the P wave, so therefore the atria would contract sometime after the P wave but before the QRS complex. So if the sinoatrial node is not functioning correctly, the atrioventricular node will set what is called junctional rhythm. So notice in this electrocardiogram there are no P waves, since the sinoatrial node is not depolarizing in order to create those P waves. So in this instance, the AV node is what's setting the junctional rhythm and the overall rhythm for the heart itself. And you'll notice that junctional rhythm is quite a bit slower than the SA node rhythm, and the AV node rhythm sets the heartbeat at anywhere from 40 to 60 beats per minute. So it is quite a bit lower than the sinus rhythm that is set by the sinoatrial node. So as the ventricles begin to depolarize, which is caused by stimulation of the ventricles by the AV bundle and the bundle branches, this is going to create the QRS complex in the electrocardiogram. If the AV node fails to conduct some of the impulses which are sent by the sinoatrial node, this is referred to as second-degree heart block. Second-degree heart block usually appears as two P waves for each QRS complex. There is one P wave in the correct position where it should be, which is immediately before the QRS complex, and then there's another P wave immediately after the T wave with no QRS complex following it. There are multiple causes of second-degree heart block, and these causes include reversible ischemia, which is reversible periods of low blood flow to the heart, typically due to things like atherosclerosis of the coronary arteries, myocarditis, or increased tone of the vagus nerve. So that means if the parasympathetic division of the body is highly active. There are even medications that can slow AV node conduction, like beta blockers or calcium channel blockers. These drugs are designed to treat things like ventricular arrhythmias by slowing nerve impulses to the heart. So resuming our discussion of the waves and deflections of the electrocardiogram, we see that the ventricles will contract immediately after we see the QRS complex. But again, in the electrocardiogram, we don't see the ventricles contracting. We only see the electrical activity of the ventricles. Atrial repolarization is also present, but it is not visible because of the large amount of electrical activity from the ventricles, and that electrical activity essentially masks the voltage displacement that would be seen by the repolarization of the atria. So the atria do repolarize, and there is an electrical event that occurs during atrial repolarization, but we simply can't see that in a typical electrocardiogram because of essentially how electrically noisy the QRS complex is. Finally, we have the repolarization of the ventricles is going to create the last deflection in the electrocardiogram. That's called the T wave. So as we have just seen, the heart is capable of generating its own depolarizations and contracting rhythmically due to those depolarizations. The nervous system, specifically the autonomic nervous system, does have connections to the heart pacemaker cells and are therefore able to modify the heart rhythm depending on the situation. So the autonomic nervous system cardiac centers are specifically located in the brainstem, the medulla oblongata of the brainstem to be even more specific. The first of the cardiac centers is called the cardioacceleratory center or the cardioaccelerator center. This is part of the sympathetic division of the autonomic nervous system, and it functions to increase the force and heart rate, or the force of contraction as well as the heart rate. The second is called the cardioinhibitory center, which is part of the parasympathetic division of the autonomic nervous system. And impulses that are firing from these nerves will tend to cause heart rate to decrease by stimulating both the sinoatrial node and the atrioventricular node.